This video reviews red blood cell structure and function. We'll start with the functions of red blood cells. The red blood cells transport our oxygen to the tissues. They also function as carriers of carbon dioxide, and they participate in acid-base balance. Red blood cells do not have mitochondria, and so they rely on a steady supply of glucose for their metabolic needs. The red blood cells are the most common type of blood cell. A mature red blood cell is called an erythrocyte. It's a non-nucleated biconcave disc, and this unique shape of the red blood cell contributes two different ways to the oxygen transport function that is the primary function of the red blood cell. One, this biconcave shape provides a larger surface area for oxygen diffusion than if it were a round cell. It's also flexible, so it has this flexible membrane that allows the red blood cell to squeeze through our very small capillaries. The red blood cell is thin. This allows oxygen to diffuse rapidly across that cell. Most of our oxygen is carried bound to hemoglobin. So 95 to 98% is bound to hemoglobin. In fact, oxygen is poorly soluble in plasma. A hemoglobin molecule is composed of two pairs of structurally different alpha and beta polypeptide chains. Each of these four polypeptide chains consists of a globin and a heme unit. So each molecule of hemoglobin can carry 4 molecules of oxygen. Hemoglobin is a natural pigment, and it has a high iron content. In fact, iron is required for the synthesis of heme. And so when oxygen is attached to the hemoglobin, our blood appears red. And when the blood is deoxygenated, it has a bit of a bluish tint. The rate at which hemoglobin is synthesized depends on the ability or the availability of iron to make this heme or the heme synthesis. The amount of iron in the blood can range from 2 grams up to 6 grams. And body iron is found in several compartments. Most of our iron, so about 80%, is complex to the heme and hemoglobin. There are small amounts found in myoglobin, in the muscle, and the cytochromes, or the enzymes that contain iron. The remaining 20% we store in the bone marrow, in the liver, in the spleen, and in other organs. We get the iron from our diet. We get it from heme sources such as meats and eggs, and we get it from non-heme sources such as beans, tofu, spinach, and nuts. This dietary iron can help maintain our iron stores. In the plasma, or in the blood, iron is transported as transferrin. So transported transferrin. In the tissues, iron is stored as ferritin. Our bodies are very efficient and eco-friendly, and so as the red blood cells age, we recycle all of the components, including the iron. The production of red blood cells is called erythropoiesis. Our red blood cells are produced in our bone marrow by what we call pluripotent stem cells. These pluripotent stem cells can then become erythroblasts, or precursor cells. red blood cell production, or erythropoiesis, is stimulated by a decrease in blood oxygen content. Our kidneys are able to sense this. Our kidneys have several endocrine functions. One of them is releasing erythropoietin in response to decreased oxygen content. Erythropoietin then goes to the bone marrow to stimulate proliferation and maturation of this precursor cell. Our immature red blood cells are called reticulocytes, and these mature erythrocytes are what we call our red blood cells. A red blood cell lasts for about 120 days or about 4 months. Every day we replace 1% of our red blood cells. So as you are listening to this, your body is busy making some new red blood cells. Most of the heme, when we recycle a red blood cell, is converted to unconjugated bilirubin. This is what we call plasma insoluble. It is sometimes called indirect bilirubin. When this unconjugated bilirubin accumulates in the blood, this is what causes a yellow tint to the mucous membranes and sometimes even the skin that is called jaundice. This unconjugated bilirubin must go to the liver. The liver processes it and turns it into conjugated bilirubin. This conjugated bilirubin is secreted in our bile, which goes into the feces or urine, and that's because it is now water soluble. Conjugated bilirubin is also known as direct bilirubin. There are several tests that are important in looking at red blood cell numbers and function. You can look at the number of red cells in the blood, and so this would be our red blood cell count. If you look at reticulocytes, you can look at the rate of red blood cell production. You can also measure hemoglobin and the amount of hemoglobin in the blood. Another measure we use is the hematocrit. This is the volume of cells in our blood. So a normal hematocrit is somewhere between 37 and 50%. When you have a low proportion of red blood cells, that would be something like anemia. A higher hematocrit might be something like polycythemia, where you have excess production of red blood cells. And you can also see an increased hematocrit with dehydration. We can also look at the size of the cell and looking at the mean corpuscular volume. We can look at the concentration of hemoglobin in the red blood cell, and this is the mean corpuscular hemoglobin concentration. And then we can use the mean cell hemoglobin again to look at the red cell mass. So these are common lab tests you will see related to red blood cells. And that is the end of our quick look at red blood cell structure and function.